cancer cell is dividing, and then this enzyme comes and adds new base DNA to the end. The cancer cell is again dividing, and new base DNA is attached to the end. If you increase the lifespan, you also increase the cancer risk. There are many Earth-like planets. We see more and more. The logic of chemical synthesis is the same. The rules are universal. And so I believe there must be something.
You are also engaged in the study of new tRNA bases. And as we know already, this may also contribute, this may be a new solution to cancer diagnostics. Maybe you can say a few words about it.
Yeah, so first of all, we look at tRNAs because tRNAs are full of modified nucleosides. So tRNAs contain, of course, the standard, the canonical nucleosides, A, G, C, and U. But in addition, more than 150 chemically modified nucleosides are known to exist in tRNAs. And this is a huge diversity. And this diversity, chemical diversity, establishes a second layer of genetic information, if you wish. You have the sequence information that allows base pairing, and then you have these additional chemical modifications on all these nucleosides. And the first question is why? Why are these tRNAs so heavily modified? What is the reason for it? Then the second question that brings us back to prebiotic chemistry is, are these modified bases new developments that nature developed late during evolution in order to cope with higher complexity? Or were these molecules already companion molecules early on? We call them vestiges of an RNA world, and that have simply survived. This would be chemical archaeology, right? So are they remains of an old, a more diverse RNA world where RNA decorated itself with peptides, where RNA catalyzed chemical reactions, and then these modified nucleosides kept their function and were later incorporated into RNA for one reason or the other. Yeah, so this is a complex field. It touches cancer research because many of these today present modified nucleosides are very important for the cell to function. We see that in cancer cells, the modification patterns are different, likely because the modification, the biochemistry to introduce these modifications is very complex, and the cancer cell is losing a lot of genetic material information, and they may not be able to modify the tRNA properly, or they don't need the modifications anymore. I don't know what the reason is, but we see differences. The modification pattern between the cancer cell and the normal cell is different, and this in principle allows to establish a new diagnostics. Yeah. So that's one aspect. The other aspect is, of course, the evolutionary aspect that interests me heavily.
What's about this aspect exactly?
Yeah, the question of whether were these modified nucleosides late developments that came late in evolution, or were these modified nucleosides companion molecules that were formed together with the canonical Watson-Crick nucleosides A, G, C, and U as companion molecules. And I believe that many of, not all, but many of the modifications that we find today in our tRNAs are indeed vestiges of an old messenger RNA world. And in fact, the growth of peptides on RNA depends in part on these modified nucleosides.
Okay. Theoretically, can DNA editing make us immortal by putting us just simply out of aging process?
Oh, aging is a super complicated phenomenon. And everybody wants to control aging, right? Because we all want to live forever. Well, one thing is clear, cancer cells are immortal. So cancer cells kill us, but they are immortal. As long as there is a human being that can host it, they are immortal. I can take cancer cells, and the whole cell biology today depends on cancer cells. Because if I take cells out, these are so-called primary cells, and keep them in culture, after 3, 4, 5 cell divisions, they are dead. So all the cell culture work that is done around the globe is done with cancer cells, because I can keep them in culture. They grow forever. They are immortal. And why are they immortal? This has to do with an enzyme that is called a telomerase, because if you want to copy a genome, the copying enzyme, the DNA polymerase, has to dock at the ends of the chromosomes. It has to dock onto the DNA, and then it starts to copy. But the position where it docks cannot be copied. So with each copying, you lose at the ends a little bit. The protein docks here, the protein docks there, and then you lose information that cannot be copied. You can copy just at the beginning. So you copy, but the position where it is docking to DNA is not copied. So you get a reduction of chromosome size with every cell division. And this is a natural barrier for immortality. Yeah, I mean, these chromosomes get shorter. At some point, important parts of the genome are reached that are not copied anymore. The cell has to die. And what is so interesting is that nature, of course, knows this, and this is why at the end of the chromosomes we have so-called junk DNA. We have repetitive elements, so-called telomerase, telomere ends, which are repetitive, GCT, 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 so they can be lost. So this is waste DNA, if you want to. And this waste DNA is consumed up with each cell division, and at some point there is no waste DNA anymore, and then the polymerase moves into these coding regions, and then the cell has to die, because important parts are not copied. And cancer cells have an enzyme called telomerase, which constantly extends the ends, these waste DNA that is complemented. So the cancer cell is dividing, and then this enzyme comes and adds new waste DNA to the end. The cancer cell is again dividing, and new waste DNA is attached to the ends. And this process, which the enzyme that does this addition of waste DNA is called telomerase, and this telomerase makes cells immortal. Yeah, that's the trick of why cancer cells are immortal.
So theoretically, it is possible.
Yeah, but then the whole body would, all the cells would constantly divide, and we would be just one big blob of cancer. Yeah, and this is indeed a problem. Yeah, this is so the cancer and aging are highly related. So yeah, human beings are fine-tuned, are tuned to have a certain lifespan and to have a relatively low cancer risk. If you increase the lifespan, you also increase the cancer risk already at the beginning of your life. So at the moment, we are tuned to have a very low cancer risk until we are about 40. Of course, there are people who have cancer with 40, but in principle, we are tuned to have low cancer risk until we are 40, because that's the reproduction phase. And then the cancer risk is slowly increasing, but it's staying low until we are 40, and then it's increasing. And it's increasing at the end with 90 or 95, you cannot avoid cancer anymore, basically. And that defines the lifespan. So if you increase the lifespan, you also increase the cancer risk early on during your reproduction phase. And so this is the balance we are living in. So if we increase the lifespan, many people believe that we pay for it with a higher cancer risk when we are young.
How did it happen in the course of evolution that all amino acids have basically the same chirality?
Yeah, that's another major question that nobody has a good answer for it. So what we know is, we know why you need homochirality. So we know why all amino acids have to have the same chirality, because only if they have the same chirality, they can form into stable 2- and 3-dimensional structures. That's also true for nucleic acids. You need to have the ribose has to be always in the same, it has to be the same enantiomer, D in our case. Then the corresponding homochiral strands can form duplexes. So for DNA and RNA, you need all the sugars have to have the same chirality in order to base pair. In peptides and proteins, all the amino acids have to have the same chirality in order to fold into stable structures. So you cannot mix chirality. You cannot take LD, LL, DDD. These things are spaghetti and not folded structures. And the same with the sugars. You cannot have LD, LD, LD mixtures. They will not base pair. So we understand why the word is homochiral. But for the amino acid, it can be either homo L or homo D. Both is fine. Organic chemists have already synthesized enantiomeric proteins. They are super stable, super fine, no problems. We can also replace the D-ribose by L-ribose and can make the mirror image of contemporary DNA and RNA. That's also fine. So I can explain why we need homochirality, why everything has to have the same configuration, but I cannot explain why the amino acids are homo L and not homo D, and why the oligonucleotides are homo D and not homo L. And there is a world thinkable, absolutely, it will be the same world, a chiral world, where you have, where the amino acids are and the sugars have a different configuration than what we see today. Such a world there is no argument at the moment that such a world cannot exist. Yeah, that's the bitter truth. So maybe you exist as a mirror image on a mirror Earth planet at the other side of the sun. This was I think a science fiction movie or story years ago, that there is a mirror image Earth behind the sun that we have not detected so far, which is of course not true, but it's
another secret for science. Why don't evolutionary biologists find the remains of kind of failed mutations? Why we find always the mutations which turn to be viable?
Well, you can only find fossils from organisms that lived. If a mutation occurs, typically also in the reproduction of humans, the woman undergoes an abortion. So many fertilized egg cells are simply removed by a normal abortion within the first 3 months. So a woman is pregnant, and then all of a sudden the period is coming back, and there was a little abortion. So this happens quite often. So I mean, if mutations occur that are devastating, we are then this organism cannot live, and we have no fossils. And on the other hand, I mean, if you look around, there are many people that are disabled people. You can see them, young babies born disabled, and of course these are mutations. And we have fossils from such disabled species, but of course they are rare, and it's already rare to find the fossil, so it's even rarer to find a fossil where such a disease can be diagnosed. Yeah, so it's a rare event.
Do you believe in panspermia hypothesis?
Can you explain this? Transpermia?
Panspermia is like, could life have been introduced from outer space and not originate on Earth itself?
That's how you define transpermia. So you say life, there is an outside intelligence that have deposited life on Earth. Yeah, I mean, everything is possible, but it just shifts the problem then into a different time and a different space. Life must have originated somewhere. So if you say it did not originate on the Earth because it was brought by an outside intelligence to Earth, then the question is, how did the outside intelligence evolve? So, I mean, there was a Big Bang, so there was nothing, and then there was a Big Bang, huge amounts of energy, far too much for any organic molecule to survive. Then the stars cooled down and planets formed, and there was no life in the universe at this moment in time. And then life had to form. If it didn't form on Earth, maybe on Alpha Centauri, and then it was delivered to Earth. It just shifts the problem to a different system. Yeah, but the question is still. So I don't like to make questions more complicated than they are. We should look at how did life emerge on Earth, and we should look now with this new exoplanet research. We should answer the question: under which conditions can which molecules form? And then we can tell the physicists, and when they say, okay, this is an exoplanet, it has this size, this mineral composition on the surface, this is its atmosphere. Thomas, which molecules can form? And then we organic chemists need to give an intelligent and well-prepared answer.
What do you think, how much time we will need to overcome this limit which we currently have?
I don't know. I mean, we know that the Earth is roughly 4.5 billion years old, and we know that the Earth is relatively young. So there are other planets. So if you look at the whole universe and you ask the question, okay, here was the Big Bang. This is where we are now. Our Earth is 4.5 billion years old. So when did the first planets form that can sustain life? The estimate is between 6 and 7 billion years ago. So there are planets on Earth, in the universe where life had 3 billion years more time to evolve. Yeah? And the big question is, okay, if life evolved there, these species are 3 billion years older than us, which means we take us and we go 3 billion years into the future. Yeah? So this is a huge amount of time. I don't know what's possible in 3 billion years. And this is why I'm always asking, where are they? Yeah? Where are these organisms that are 3 billion years older? So maybe they are around, and we are simply too stupid to see them. Yeah, but so this is the dimension in which we have to think. Yeah, it's not 10,000 or 20,000 years. We have to see that potentially there are organisms, species that had 3 billion years more time to evolve.
Maybe for the final question, do you believe that there is life on other planets already?
Well, I wouldn't be surprised. First of all, there are many Earth-like planets. We see more and more. Yeah, exoplanet researchers got a Nobel Prize a few years ago, and we will see more and more exoplanets with the new James Webb telescope. And I believe that on all these planets, the logic of chemical synthesis is the same. The rules are universal. Physics is universal. And so I believe there must be something. Is life as complex as the one here on Earth? I don't know, but I think life forms will be found. I'm relatively sure, and there are strong limits to intergalactical traffic. We know that it's almost impossible to reach other solar systems, yeah, Alpha Centauri. So yeah, I think I'm relatively optimistic that in the future mankind will find that they are not alone in the universe.